All right, well, after that somewhat debacle, thank you everyone for your patience, and I'm so glad to be here with you tonight to speak to you about what I've learned about death adders, and I'm calling them the black sheep of Australian snakes because they're so unique. So I was told to introduce myself, so I'll spend just a couple minutes doing that. I'll pronounce my surname for you, Christina Zdenek. And if people who are listening online could mute this themselves, that would be greatly appreciated. Video is fine for you to keep on. So where it all began is basically with a complete fascination and love with reptiles for as long as I can remember, and I'm sure a lot of you are much the same. They're just a spectacular taxa, and this is me when I was 11 years old, and by then I'd used all my savings to buy a pair of chameleons, and I bred them, and here are all the young sitting on my hands when I was about 11 years old. Just constantly fascinated by reptiles, had many different sorts as pets. And of course, you can't have chameleons in Australia. So I didn't grow up in Australia, actually, Los Angeles. And so, yeah, I want to acknowledge too that I definitely had a privileged upbringing and supportive parents that allowed me to get to where I am today. So I spent a lot of time researching palm cockatoos, which I know don't have much to do with death adders, but I'm a bit of a bird nerd. And so they're up on Cape York, and they have the great habit of drumming. So I spent about 7 years, total of 28 months during the dry season studying that bird. And that was through the Australian National University. And I also joined a koala project for a while where I was collecting leaves to sample. So a lot of field work, just involved with a lot of different Australian flora and fauna. Also, I do a fair amount of work, like nature documentary work for various different companies over the years. So it takes up a bit of time, but what I'm mainly paid to do, my daytime job, is I'm a lab manager and postdoctoral research fellow at the University of Queensland. So we're here in Brisbane, that's where I'm at the moment. And the lab I'm part of is the Venom Evolution Lab. So we look at snake venoms and their function in human body, and also in pets, and anti-venom efficacy as well. So let's get down to it. In Australia, we are a very lucky country in that we are the land of reptiles. We've got 105 highly venomous snakes, land snakes that is. Here I've got a beautiful coastal taipan photographed by Chris Hay. We've got, of course, the red-bellied black snake. These are common species that people encounter around creeks. Tiger snakes, mainly in colder areas. And of course, the infamous eastern brown snake, very common throughout suburbia. Gavin Smith, I'm sure I'll tell you plenty about them. And of course, the death adder. So this one's a common death adder. This one I photographed on Magnetic Island. And a fascinating species that I'm going to tell you all about. So death adders generally occur throughout most of Australia, not in Tasmania though, and not a lot in Victoria, but the rest of Australia, you'll get a species of death adder. And if we sort of plot them around Australia, this is roughly where they are. Here's the 7 species that are currently recognized within Australia. And then we've got Acanthopus laevis in New Guinea, and there might be a few other species in New Guinea as well that just simply haven't been identified. Now, this is a pretty broad overarching perspective, and you can't really see the borderline. So what I've done is made this map, courtesy of Atlas of Living Australia, whereby you can see actual observations of these species, and you can see the species list on the right there. So located throughout Australia, basically in patchy environments. They're pretty hard for herps to find. I'm sure I don't need to tell you guys that. They're cryptic species. And if you look at their relatedness between the species, so Wolfgang Worcester, Chris Hay, and Brian Fry, and a few colleagues in 2005 came up with this paper, published this paper called Snakes Across the Strait. And these concentric circles indicate relatedness, and sort of like a topographical map in a way, looking at the relatedness. So you can see the New Guinea population quite different, Northern Territory and sort of Cape York a bit different, and then the East Coast and the South more related to each other than the others. Now, if you look at the order Serpentes, snakes, there are 21 different families of snakes in the world, and Elapidae is where death adders sit. So they diverged around 35 million years ago from sea snakes there, and that's a timeline shown there. And there's 9 families of snakes which contain venomous species within them. So within Elapidae, so why have I called Australian snakes the black sheep? Why have I called death adders the black sheep of Australian snakes? Well, if you look at some examples, common, the coastal taipan and the eastern brown, they are active foragers. They will go in quick pursuit of their prey, whereas death adders, as you probably are aware, are an ambush predator. They're the only true ambush predator in the family Elapidae in Australia. So here's the beautiful death adder, a typical one. This one's from Weipa. I photographed it up there when I was going to collect some venoms. And so as you can see from the shape, they got this stout, broad body shape and a sort of rat-like tail. It's much skinnier on the tail and a triangular head. So because they're in Elapidae, they've got fixed fangs. And they also have a highly evolved tail that's specific for caught alluring, to lure their prey to them. So it's super dexterous, very fine scales at the end. So a true ambush predator. And if you compare elapids to Viperidae, so vipers around the world, you actually have an example of convergent evolution, whereby death adders and vipers have that triangular head and also sort of skinnier rat-like tail. And they have sluggish movements, but when they strike, it's a rapid strike. And so they're sort of like, for any handlers, you might have heard of the death dome, where you don't go anywhere near the strike of the snake, so within that death dome, because before you know it, you're bitten and you can't even move quick enough. So they diverged from each other, Elapidae and Viperidae, about 60 million years ago. So that's how different, but despite that, death adders have really converged into more like a viper snake in Australia. So making them quite different from other elapids, which I think makes them extra fascinating. So when you compare their fangs, so Elapidae fangs are fixed, so they're not able to be moved. Whereas here, this Russell's viper that Chris Hay's holding, they have the hinged fangs. So that's a feature of viper snakes. But they both do have long fangs, and compared to its body size, death adder have quite large fangs. And their head actually moves. When you hold them in the hand, they can almost do this movement with their fangs, which is similar to a viper being able to have that mobile fang action. And so here it shows, when they're underneath, and then when they go to bite, it actually extends outwards, and they've got structures to support that. So death adders, the conservation threats, I figured I'd start with this aspect. These are my assessments of what I think are the main conservation threats. And we don't have a heap of information on these specifically with death adders, but I believe number one would certainly be land clearing. And I'd say that be the case for almost all reptiles in Australia. Second, being unnatural fire regimes. So being very frequent and very hot fires, for example. So death adders, they hide in the leaf litter, not only for camouflage, but probably for thermoregulation as well. And so if a hot fire comes through and there's no more leaf litter, this would be a serious issue for death adders, probably leading to their demise and death in an area. And then, of course, pest species like the cane toad, also foxes and feral cats. So all in all, yeah, bit of a dire situation, certainly in some places. So the conservation status is, not a lot of data going into a lot of the species of death adders, but there is one listed as vulnerable, and that's the common death adder, ironically called common, but it's listed as vulnerable, and all the rest are least concern. But again, data deficient really would be a better way to describe it, I'd say. So we do have some information about the influence of cane toads on death adders, not from my work, but from previous people's work, which I'll go into. So they were introduced by the Australian government in 1935, as you're probably aware, to hopefully eat cane beetles. And they then spread throughout Australia, decimating birds, snakes, and lizards, monitor lizards, a whole suite of species. And that's because when they're attacked, they exude a poisonous toxin from their parotid glands behind their eyes, leading to the death of the animal that's trying to eat them. So some animals, fortunately, have learned to turn the toad over and can eat there, and it's no problem. Like Ricali, I guess a paper recently came out about that, and some birds. Okay, so I thought this was really interesting to show. So if you think about the impact of cane toads on death adders, so a lot of death adders eat frogs. This is a really great map by Mark Urban showing the rate of cane toads, the way they've traveled across Australia. So originally moving only about 10 kilometers per year. So down the bottom here in blue, so across this region. But as they've moved further west, they've actually progressed quicker and quicker every year. And that's because the cane toads at the front line are breeding together, and they're actually changing physically, even getting things like arthritis because they're just moving so much. But those ones breeding together and being able to travel further again. So evolution in action. So this means that more populations of death adders could be at risk from eating cane toads and dying from them. So fortunately, Associate Professor Ben Phillips years ago studied death adders in the Northern Territory, particularly in Fog Dam, and garnered some really interesting information about death adders. Here's an image that he took of one eating a cane toad. And he found here in a place called Adelaide River Floodplain, 60 k's east of Darwin, that as you can see from these two graphs, which is the mean death adders per day that was seen, it's going down over time. And in that same time, cane toads going up. So there's an inverse relationship there. I can't really do it, but yeah, as you can see, the more cane toads, basically, the less death adders. And so it seemed like it was a pretty clear story that death adders were dying because they more cane toads, and that's the prey that they were taking and then dying because of that. So that was a bit of a worry. And then interestingly, Brown et al. Came out with a paper whereby they showed that praelongus was actually, it increased after cane toads arrived, and this arrow indicates when they arrived here at this location. Yeah, in 2005. So why that was the case is not quite understood, but it could be that a predator of death adder, a death adder in that area, that species, was taken out by the cane toad, thereby allowing sort of like meso-predator release style. Shazad, if you wouldn't mind muting your mic, that would be greatly appreciated.
Sorry.
So this led me to start the Death Adder project. This was in about 2015. So fortunately, I had some freedom during my PhD where I could study what I was really interested in. And so I started this project because I was just fascinated by death adders and how they were so different from all the other Australian snakes, and that we seem to know so little about them. And so I worked for, I don't know, a year and a half to try to win grants to study them. Finally cracked a couple. I won a grant from National Geographic and also Australian Geographic, and then got some support here and there. And then basically another like 6 months to get animal ethics and a lot of training. A lot of things came to play and a lot of hard work, but it allowed me to go to Magnetic Island, whereby it's well known for a decent, healthy population of death adders. If you want to see a death adder and you haven't seen it on mainland Australia, you're told as a herp, go to Magnetic Island. And it's an interesting place, a very beautiful place. And you've got this urban bush interface where you've got death adders in the bush, but encountering people. So I was interested too in the human snake conflict, but in addition, the pet snake conflict. So if you look in Australia, sort of Australia-wide, from snake bite, about 2 to 5 people die per year, but pets is about 213 deaths. Now that's to be a way underestimate, because a lot of pets that die and people don't know why, they don't bring them to the vet, so it's not counted. And if you compare that to Magnetic Island alone, in just 1 year, there were 10 dogs and 2 cats that died from snake bite. And the vets really do think it's death adders based on the symptoms that they were presenting with. So it was a particular pet snake conflict going on in Magnetic Island. I was hoping to understand death adders a bit better there to hopefully ameliorate this issue. And if you look at other studies of relocating snakes, this is generally the common practice for reducing that human snake conflict. Somebody rings a snake catcher, like Gavin, Eastern Browns in Canberra, and they remove them away from people. Nick Clement in 2004 published a paper about the relocation of tiger snakes and found that once they relocated those animals, their movement patterns were quite erratic. Their home ranges became larger. And so if you think about it, maybe that's not the best solution if you might have a greater chance of encountering a human, because you're just moving all over the place and having a larger home range if you're that snake that's been relocated. And there's been some papers coming out about king cobras. They need obviously much larger home range, having a lot of conflict with humans there. But the general consensus is that it's a negative effect on the snakes when you translocate them. But because death adders are such a unique species within the suite of Elapidae in Australia, I wanted to see how death adders in particular responded to translocation. And so
early
here's
the next
a video.
Day, another death adder report, this time deep in the bush.
So we're on a death adder call out. They've got it at their house. It's sort of like a bush setting, and we're heading out to the more remote side of the island. There's no water or power. They're off the grid here. And so hence it's a dirt road, and we need a bit more of a four-wheel drive vehicle to do that.
And on our way to this remote site, I couldn't help but think about this human-wildlife conflict. Here's an ambush predator who lives life hunting small mammals and reptiles. It prefers large tracts of wilderness like we have here on Magnetic Island. But when we build houses and we bring our pets, there is inevitable contact. And unlike most creatures, this one can deliver a lethal bite. How can we live in tandem with these death adders? The key, it seems, is learning as much as we can about them. And that's why I'm so excited to follow Christina's research.
All right, so here is a snake call out, and this is a death adder. Have a look at how flat this thing gets. Very cool behavior.
And then another big part of our research was milking them for their venom. Venom, as it turns out, can, in small amounts, actually be used to help treat cancer patients or stroke patients. It's a medicine. And this is a big part of her research and a huge reason why she wants to protect these snakes and to change the public's view of venomous snakes.
If we killed all the venomous snakes in the world, you'd be losing that ability to extract the venom and use it for life-saving medications.
Okay, so it's a short clip from a longer video, and if you check on my website, you can find the rest of it. But it's just an example of that conflict I was just describing. So finally got to Magnetic Island, went there a few times, and here is a sort of a newspaper clipping from a local newspaper on Magnetic Island, whereby it shows the work that I was doing there, whereby I could actually just talk with a heap of locals and find out where and when they encountered death adders. And so I thought that was a really good source of being able to chat with the locals and understand their interaction with snakes, and then try to plot that. And thanks to the help of some great volunteers that came along for some of my work, I was able to put together this somewhat raw but informative graph of the number of death adder encounters on the y-axis, and on the x here we've got all the months across the year, and these are the sheer number of incidents that people encountered death adders. And I know you're probably thinking that snake identification can somewhat be dubious with the general public, but a lot of these people did have images that I could confirm that they were death adder encounters. So yeah, as you can see, there's a couple different peaks there at different parts of the year. So this can help inform people to make sure to, you know, wear shoes going out at night and bring a torch, and just simple things like that, or don't let the dog go out at night for a pee, have them go earlier, those sorts of things. So I think it's important information for people to have. Julia Riley in the States studied a Massassauga rattlesnake and tracked them using this technique called the subdermal stitch method. And this is an important improvement and technological advancement of being able to track snakes. So a lot of animals are tracked via putting a tracker, you know, and attaching it to a limb or the neck or something like that, but you can't with snakes. It's more difficult, right? But she was able to attach it to their tail safely, and she found no problems with doing that. And the snakes had it on for, I think, 6 to 9 months. And so that gave us hope that rather than having 2 surgeries to implant a transmitter inside, to surgically implant them internally, which could be very expensive. I was quoted $1,000 a day for a vet, and max 4 surgeries a day. This was a good cheap alternative. The radio transmitter is only a couple $100, and after I was trained by a vet, I was able to do all these attachments myself. So this is a paper that just got accepted a couple of weeks ago, which will be coming out soon, whereby I use this technique on death adders in order to track them on Magnetic Island. And so I got a suite of volunteers together, and because, as you know, death adders are very hard to find. So we had a super hard task ahead of us. But Elliot Budd, Daniel Bromley, and Daniel Dashevsky were fantastic volunteers for us, worked hard every day. And Chris Hay and myself co-led the project on the island to find and track death adders. So to give you a perspective of what the habitat looked like, here is a snapshot that Chris took. So it's almost completely closed canopy, not rainforest though. Some light does get in, but it's pretty, there's some vines in there. And as you can imagine, extremely difficult to find death adders. There's all that leaf litter, great rocky outcrops, and a few dry creek beds throughout the study site. So this doesn't represent all of Magnetic Island, but the place that we were working, this is what it looked like. Okay, so pop quiz, spot the death adder. Pretend you're out on Magnetic Island. Everyone loves this game. And try to find the death adder. Now, I'm really narrowing it down for you, not a whole hour-long trek or several hours. Now, raise your hand if anyone spotted the death adder. No one's coming close. Oh, it's hard. Yeah. If you want a hint, you guys ready for a hint? I've zoomed in on it for you. Now, can you find it? Still can't find it. Yeah, this is what we had to deal with. So I hope you can you realize how difficult our job was. There it is there. So if you can see, there's a couple of bands there of the death adder. So it's on the move going upwards there. So super hard work, hey. Even when we were radio tracking them and knew they were within, you know, a few square meters, it would take upwards of a half hour to find them. And you, of course, you don't want to step on them and you don't want to get bitten. So precarious job. But we did find 10 of them in just a couple of months. So well done us. And interestingly enough, only 2 of them were found by road cruising. The rest, 8 out of the 10, were found on foot. Now, unfortunately, only 5 of these were large enough to attach the radio transmitter to, even though it's only 1.5 grams. And this is one interesting part that came out of the research, how small death adders are on Magnetic Island compared to the mainland. That was really actually quite interesting. So these ones we were able to track for up to a month, but we did it daily. So we got quite a lot of data. So once we did finally find a death adder, we take them back to the research station, Amaru and Mandalay it was called, generous sponsor of the project. And Chris there's got a snake tube, and I'm coercing it into the tube so that we can safely handle it and perform a very short 15-minute procedure with 2 small insertions there, kind of like a piercing, if you like, a bit of a thicker piercing. So it's the same size needle as like if you were to get your pet dog inserted with a microchip inserted into their neck. That's the same size needle I was using. But of course, first putting some local anesthetic there with an insulin needle, and then tying on the transmitter to the top of the tail. And so here's a finished one with Chris and I. And so the next stage was to put them back where we found them, or if we got them from a house, that's a translocation, and we need to find a suitable place, and then come back the next day, and then subsequent after that every day to try to find them. And here we are using an antenna with an individualistic radio frequency to try to relocate them. Marking the tags, going out at night, so morning and night hikes. And here, fresh data here for you, never been published before. And for the paper, it might look a bit different, but here's just sort of like the raw plotting. If you look at the y-axis, you've got the distance traveled. Now that was since the last time that we marked where they were. And then we've got on the x-axis, the days since release. So this one was tracked 26 days. Her name was Madam. And as you can see, there's some spikes along the way, but no more than about 6 meters did she move. Now, I'll then go and show you all the resident snakes on top of one another, now that you're familiar with the graph style. And they've each got a different color. So here's a boy in green, another girl in red. She was tracked for a much shorter time period. But again, sort of similar patterns. And this one, again, maps 6 meters. So not too surprising. Death adders aren't known to be, moving a lot, but you never know until you really do the work. And how about the translocated snakes? So these are snakes that people rang us, said, come get the death adder. And then we removed it from the house and chose a suitable place with good shelter in the bush. And an interesting point here with Granny, she had a really big spike. So we dropped her off on day 0. The next day, she had moved 16 meters from that spot. So presumably she wasn't quite happy with the spot where she was, trying to evade something or find good shelter, not exactly sure. But then she seemed to settle down, bit of movement here and another spike. I think that was about 12 meters she moved. And she found a really good rocky outcrop and then stayed there thereafter. Soldier, he was a translocated snake. He didn't have the big spike. He seemed to be happy where he was. And then he pretty well stayed there for, I think it was 10 days that we tracked him. So that is all of them overlapped over the top of one another. And I think the one that really stands out to me is Granny, a translocated snake. And if you compare, okay, about 6 meters, the other one, this one's a bit twice the distance she's traveled. So maybe that translocation has had a negative effect on her. You kind of have a true control of having the same individual be, a resident and a translocated snake at the same time. But I suppose with more snakes, you might be able to get a better idea. But it is super hard work. It took years just to get this. We did our best. And a lot of translocation papers only have very small sample size because it's an immense amount of work. And it took 3/4 of the day to find all these guys on a daily basis. Okay, and at this stage, I'd like to change it up just a little bit and introduce Chris Hay. He is a well-known herpetologist, has been so for 30 years. And he actually is one of the well-known, well-known people who know a lot about death adders. And he did a lot of the field work about death adders for Snakes Across the Strait paper. And coincidentally, he's also my husband. And my interest in death adders actually kind of brought me to get to meet him. So without further ado, I want to introduce you to Chris and let him tell you about this cool story. Go ahead.
Yeah, good evening, everyone. So the slide that Christina's put up here was, it sort of illustrates something that we noticed quite a lot when we were finding these snakes on a daily basis, going out, tracking each one, and sitting back at a distance, not to disturb the animal, but observing it for about half an hour to 45 minutes. And it only took a few days to notice a bit of a trend forming. When we came across these snakes, more often than not, they had positioned themselves buried in leaf litter, but with the head and forebody exposed, usually sitting on a rock surface that was either vertical or sort of on a bit of a gradient there. And we kept seeing this over and over and over again. We started to think, what are these snakes doing? Why are they continually doing this every day? And it was one particular day we were out tracking our snakes and we got the answer to it. We actually observed these death adders, they're diurnally hunting. Now we think of death adders, yes, they're a nocturnal snake. They'll move from A to B under the cover of darkness. But these animals were specifically hunting diurnally because their prey, which is the various skink species on the island, are also diurnal. And we actually witnessed two feeding events while tracking these snakes. So Christina's just highlighted the red section. That is the death adder in that ambush position. Time of day of this is probably about 10:30, 11 o'clock in the morning. And in the other photo there, you can actually see a little skink, a little Lampripholus mirabilis, I believe. So that snake is essentially waiting for that skink. And we saw this on numerous occasions among the four snakes. We could pretty much guarantee that if we sat there and watched a snake with its head sitting in that position on a rock, sooner or later we'd witness a feeding event. So it was very interesting to see these guys hunting by day. Now they wouldn't move during the day. And if their hunting spot was unsuccessful, they might stick at it for a day or two. And then we'd see one of those big stretches of movement. They'd reposition themselves and adopt the exact same position again. Because the skinks were abundant on the island. And we noticed too that the skinks generally avoided the leaf litter. If you've ever been to Magnetic Island, you might know that there's big mygalomorph spiders that live in those leaves in burrows. And the skinks were observed actively avoiding the leaf litter itself. They would jump from rock to rock to avoid touching the leaves and probably avoid a predator. Well, of course, the death adders were right onto that, and they were ambushing these skinks as they were using their little rock highways to get around.
Yeah, it was so cool. Really great observation, just from, straight hard work. Here's another example. I think if I highlighted it. Yeah, so here's another example that Chris was describing. This is another snake, and here's another rock where it's angled up again, just as he described. All right, so I did promise to talk a little bit about their venom as well. So death adders, I mean, the name kind of gives it away, but they are highly venomous. And the only sort of paper we've got that tells us about the death rate before anti-venom was available in 1956 was this Tidswell thesis. And he quoted that there was about a 50% death rate. And it's from people dying of asphyxiation because their diaphragm muscles aren't able to contract to fill their lungs with air. So now the death rate would be, almost none, because as long as somebody gets to hospital, for one, there's anti-venom that can reverse that action, the neurotoxic action, because it's postsynaptic, not presynaptic, and also not coagulotoxic, which you can't reverse. But also just being intubated as well, putting a tube down the throat and have artificial breathing going on is a way to combat neurotoxic venom. So I actually looked into death adder venom as well, published this paper last year, whereby first I needed to develop this new technique of how to study neurons, but using a synthetic system. So colleagues and I, particularly Richard Harrison and Brian Fry and Sanjaya Kurupu, we came up with this technique whereby just by using synthetic part of the neuron, we were able to then attach it onto a sensor that has a vertical light probe that goes, and being able to determine when something binds to the end of that probe where we've hung off the receptor. So the toxin then binds to the receptor, and you can see the shift of light read by the machine. And so here is a really cool graph showing in two different ways the same data, whereby we really found that the neurotoxins in the death adder venom weren't very targeted at any particular taxa. As you can see, here we've got wavelength, so that's a shift of light that I described for you, and over the course of time for 120 seconds, this is called the association step in a sort of kinetics experiment. And as you can see, all these different taxa, they're hit pretty well similarly across the board. And if you compare that to the king cobra, so here we've got a maximum of 0.4 and they're all within a small range, but here a max of 0.6. And we've got a very wide range of targeting of those neurotoxins across the taxa. So it's showing that, for one, the receptors on the different taxa are slightly different, between fish, amphibian, lizard, snake, etc. Just a few amino acids difference could mean that the toxin doesn't bind as well, or not as quickly, that sort of thing. And as you can see, this major one at the top, so this is, we did the experiment in triplicate, and these are the air bars, that's why they're a bit wider. That's a snake. And as you know from the genus name, Ophiophagus, snake loving, king cobras eat other snakes. And in fact, their neurotoxins we found target other snakes, as opposed to, so the death adder being more of a generalist species in terms of its venom targeting. So venom is generally thought to have evolved in most snakes, not spitting cobras as much, but most snakes to be able to immobilize and overpower their prey.
To the registrar of the university, Harry Wesley Smith, to say one Candea camelospora. He needs to leave the country.
Their microphone. Okay, so yeah, that's a bit about death adder venom. It's a generalist venom, and if you think about it, being an ambush predator, you're sitting and waiting for the prey, wiggling your caudal lure. You mightn't have the luxury of being able to chase a certain prey, right? You have to wait to see what comes across your path. And so if you have a broad sort of venom that can work just as well across any sorts of taxa, that would benefit you as an animal. All right, back to Chris to describe what it's like to be bitten by a death adder.
Okay, so yeah, that photo there is actually a much younger version of me. That is definitely not me after a night at the pub. That was after an envenomation from a floodplain death adder back in, it says 95, it was actually 96. But the snake that bit me, she was 92 centimeters long, so she's a pretty big death adder.
That's right there.
What was quite interesting about this bite was it was the first time we'd seen myotoxicity, or toxicity of the muscles, from a death adder snake bite. And that sort of prompted Brian Fry and I to look deeper into some death adder venoms to see exactly what else may have been missed. But yeah, that was a nasty bite, guys. I was had what we call locked-in syndrome, which is total paralysis for about 72 hours. Yeah, basically, yeah, the common term is coma. On a ventilator, and it took 3 doses of death adder anti-venom to reverse the effects of that bite. So it was a pretty nasty one.
They rang your parents?
Yeah, certainly a very hard lesson to learn, that if you make mistakes with these animals, you will pay the price. But it also goes to show the effectiveness of the anti-venom and the treatments available. But yes, certainly by far that was a tragic snake bite there. Now what you're actually seeing there, the droopiness of the eyes is called ptosis. So this is total paralysis of the facial muscles, including the mouth. Couldn't form my lips to spit, so I was vomiting profusely and I couldn't get that vomit out of my mouth because I couldn't spit, so that was flowing back down into my lungs. Terrifying, yeah, terrifying experience. But it just goes to show that you know, that these really large death adders and the floodplain death adder, Acanthopus hawkei, can reach close to 1 meter 20 centimeters in length. So some of them get pretty big, and it's definitely the bigger ones that I would be very, very wary of.
Yeah.
Yeah.
And they even rang his parents when he was in hospital and told him to come say goodbye to your son because he's dying. So we're lucky he made it.
Yep.
All right. So some take-home messages for you. We'll wrap up here. Some death adder species, some death adder populations, as we said, hunt diurnally. So this is new information, and that blew our minds. So a really fascinating bit of information that we uncovered there. Magnetic island death adders, they're an insular population being on an island and being isolated for thousands of years. And they're actually quite a bit smaller than the mainland population. The max size on all the records was what, 80 centimeters? 60 yeah. Centimeters max size there, as opposed to like, you know, 1 meter on the mainland. Same species, mind you. So that was quite interesting. So death adders, I guess, not having much of a home range, what appears to be the case, and also males not engaging in male-to-male combat in order to establish territory. Those two factors may actually play quite an important role. And it seems like death adders, unlike probably some other snakes in Australia, might be more suitable than others for relocations. But of course, for the snake relocators, the snake catchers, it's important for them to put them directly into a nice bit of shelter and in shade. That's super important, so that they can go into cover immediately. We still know very little about death adders, and in fact, all reptiles in Australia, basically, and that's despite reptiles dominating the Australian fauna. So it really goes to show how, you know, how mammals and birds really are the popular fauna for people funding them. That's what people care about, but it doesn't mean that reptiles are any less important. And of course, for ecosystem function as well, they're predators and prey, which is an important point. And death adders are a fascinating species, so much more to uncover. And in fact, we have a little surprise for you here.
Yep, we'll just share with you one of our death adders, guys. So this is a rough-scaled death adder, Acanthopus rugosus, from Western Queensland. And she is 7 years old now. She's been raised from a baby. And she is now pushing maybe 70 centimeters in length. But yeah, gorgeous, gorgeous.
Yeah, she's quite big.
She is quite big.
So yeah, just to make you guys in Canberra who can't keep these legally, a little bit jealous, I guess. We've got 5 death adders. We've just showed you one here.
Yeah. She's
really good for venomous snake education shows. She does a good job.
She does.
Thank you, Chris. Just want to let you know too, if you weren't aware, on ABC iView, there was this episode on Catalyst called Kill or Cure, the story of Venom. So yeah, they did a great job there. If you're interested in Venom, I'd recommend checking that out. And also, bit of a sneak peek for you. We endeavored on a new journey and adventure, and it's called the Sound Garden Project, whereby we were interested in seeing how snakes respond to sound. And so we teamed up with Professor Damien Canduso here in the middle. He's actually the head of the school for Queensland University of Technology's creative practice, so school creative practice. And so it's a really great synergy of the arts and sciences coming together. And we had a fantastic time. It was a lot of hard work, but we're working through the data and producing the figures now. So by the end of the year, we should have that paper out. So stay tuned and watch this space there. And if anyone's interested in watching the rest of that Death Adder video or any other videos, there's my website at the top, or just Google my name. I'm sure you'll come up with it. And quick mention, if anybody's got any Iguania epsisolis, been after one for a while now, so please get in touch. And other than that, I wanted to thank you for your time and attention, and hope you enjoyed the talk and learned a bit more about Death Adders, and wanted to see if anybody had any questions. There's the acknowledgements, by the way. Thank you to all those people and all those groups.
Yeah, Christina, I have a question and a comment.
Yep.
Okay. The comment is that in regards to diurnal predation, in the early 1970s, I was working on Rattus phylicissimus on the Barclay tablelands, and in picking up our traps early in the morning after setting them overnight, we came across a death adder that had just caught and killed a wader, a small wader, a bird. So it obviously got it in daylight because it had only just got it. So there's a diurnal observation.
Thank you very much. Yeah, we might get in touch about that actually, because we might include that as another observation.
Yeah, so I'm not sure if I'll be able to give you a date. I can't remember what I don't think we collected the specimen. We just observed it and continued on our way. But I might be able to get a month on it. And the question is, the death adder bite, was there any residual effects? Has it affected your smell or anything else in longer term?
Yeah, John, it certainly did. I had disturbed sense of smell and taste for a good sort of year and a half following that bite. Unfortunately, with subsequent bites over the years, I've had permanent damage of that nerve, the cranial nerve now. So I have very little sense of smell or taste left, unfortunately. Yes. A little bit of mild paralysis persisted. If I get a little bit tired and run down, I do have a little bit of a lazy eye. One of my eyes sort of closes a bit more than the other. And yeah, that's an after effect from that particular snake bite too.
How long ago was that?
That was 25 years ago now, John.
Yeah, crikey.
Yeah, long time.
Yeah. Okay, great. Thank you.
Yeah, no worries, John.
And also from the Maritime.
I have a question. This is Janet Wild from near Wagga, New South Wales. Just a quick question, that the paralysis, the eye paralysis,
yep,
would you be sure that it wouldn't have happened without the snake bite?
Well, when I was paralyzed, that eye was far more affected. And so we saw it sort of slowly, as I recovered, it slowly began to sort of resume to normal, but never quite made it back to the exact same position. So the doctors sort of said, look, you've probably had a little bit of nerve damage there, you know, relating, yeah, to the snake bite. So no, I don't think it would have happened without the snake bite. It was definitely corresponding to the most affected eye.
Yeah, thanks.
No worries.
Pat, you want to unmute yourself?
G'day there. I had a question.
Great presentation, by the way.
Sorry,
just a quick one. How long, over what period did you track the death adders for?
Yes, that was November and December. So the longest one we tracked was 33 days. Yeah, during that time period. And it was corresponding to when we felt that we had the best chance of finding the most amount of death adders. We were fighting a heat wave. There were many days of 40 degrees, which was unfortunate, but we had to move ahead with that time period due to scheduling of many things.
Sure.
Yeah. So, I mean, it would be fascinating, of course, to track death adders for the entire year, but you'd probably need a dedicated PhD. Whereas my PhD was actually on venom. Yeah,
Do
but
you
I
think-
was a
do you
student, so I did.
Do you think there might have been a wider range of travel during, say, mating season?
Yeah, absolutely. I would say, I mean, it's hard to know how far that they might travel, but you hear anecdotally that a lot of snakes are moving. What's the time period there? It's still October.
Yeah, September,
Yeah.
October would be
Yeah.
the mating, period, I'd say, yeah.
So we were sort of just after that time period.
Yeah.
But also when they're going into maybe finding a overwintering site, they might travel a bit further during that period as well, I'd suspect.
Thank you very much.
Pleasure. Thanks for the question.
I have
Sorry there, can
another
jump in and just ask a question, please? I was just curious as to sort of what distance these snakes were being relocated at, and if there was, I guess, between the snakes that moved more and versus less, were they moved at a further distance than the other ones?
No, so they were completely different places from where we found them, and that was because we wanted to put them in a suitable habitat. It needed to be the rangers there and the public generally wanted us to be at least sort of 400, 500 meters from a house. Again, this gives us great information because we know how far death adders move. So maybe those people who are translocating death adders from people's houses don't have to go as far, which is more work for them. And they often do it for free anyway. So taking up that time away from work, it can be an issue. But yeah, we had them generally within the same site. So probably within, I don't know, 800 meter transect through an area. But we would space them out through that area so that they wouldn't encounter each other. And yeah, they were well and truly beyond the greatest distance that any one of them traveled. So there wouldn't be a correlation. I mean, I don't really have the sample size anyway to test that because we sort of like one individual, there was only, I think, 2 translocated individuals and 3 residents. So yeah, they were from the general same suburb within Magnetic Island, but yeah, it didn't really look at that.
Okay, awesome. Thank you very much for that.
G'day, I had a question. You mentioned the predation of Saxicolon skinks like Lampripholus, Maribolus. Do you have any records of them predating like fossorial skinks like Glaphyramorphus or like Pygmaeus skincus?
Great question. I'm not sure if do they occur on the island.
It's quite Pygmaeus skincus do, yeah.
Okay.
Yeah, not that we witnessed. The Pygmaeus skincus, we only actually found one. It was quite a rare find, pretty exciting for us. But yeah, no, look, we didn't see any evidence of Glaphies or the Pygmaeus skincus being preyed upon. I mean, look, that's probably due to just the how cryptic those species are. Those Lampripholus were just everywhere.
Oh, they were super abundant.
Yeah.
And also, we arrived coincidentally when the March flies arrived, and they were just in full abundance, biting us to buggery. And those skinks were just gorging themselves on that period. So yeah, they were just everywhere. There's another skink there, Carlia rhomboidalis. I've got an image that somebody sent me of a death adder eating one of those on the island. But it actually brings me to another point. We did collect, I think, something like 8 fecal samples. So another part of the study, which we haven't had a chance to look at, but is basically to look at those samples under a microscope and try to identify different, hopefully, genera of skinks, just based on scales that we might find inside the fecal samples. So yeah, we hope to look at that. We've also got a couple of dead death adders, like smushed on the road, or another one that we got off somebody. And so we can look at the gut contents there. So hopefully there'll be more to this story that we'll uncover over time.
I had a question. Sorry about it being a bit irrelevant, but what's the current taxonomic status on the magnetic island death adders, and how do they sort of fit into that whole framework with the northern adder sort of species?
Yeah, so it's kind of like right on the border of those two species, and people have been wondering, but it is antarcticus that occurs on the island there. Yeah, so it's based on the mid-body scale count. Differences between those two species, praelongus versus antarcticus.
Christina, I've got a question.
Yep.
Regarding the graph that you showed of the days since release and the distance traveled.
Yep.
Did you try relating the distance traveled to ambient temperature?
Yeah, great question. We recorded a lot more data than what I showed here tonight. So journals like to be the first ones to publish data. So wasn't able to kind of reveal everything, but that is something that we did collect and we'll be looking at. Because one of the interesting questions that we were after is what triggers their movement, you know, these spikes that we're seeing. So yeah, we'll definitely do full proper analysis. Those figures that I made was just basically showing raw data, no stats, and no correlation of various variables like ambient temperature. We also collected air pressure, which we think might be a trigger for them, a drop in low air pressure.
Are you able to say whether there was a correlation with temperature?
At this stage, no. I'd probably have to really look at it in detail.
Yeah.
Yeah. So we were working in a warm period. So, you know, if it's a cold period, like the temperatures weren't shifting that much.
Pretty consistent.
Yeah, it's pretty consistent. You really have to go across the whole year to do, you know, that proper comparison there.
Yeah. So I'm guessing Magnetic Island is a pretty equable climate.
Yes, it's lovely. Our temps in November and December were about 30 to 40 degrees.
Yeah, it was very hot. It was very hot.
Oh, yeah. A lot of the days toward the end were like, 37 to 40.
So being an equable climate, you wouldn't have great differences between days, unlike a continental climate.
Yeah, correct.
Yeah,
Yeah,
absolutely.
Yeah. Definitely a variable in the weather up there was periods of thunderstorm activity that did actually bring rain. So it'll be interesting to see if those spikes in movement line up with those thunderstorm events.
Yeah. And then yeah.
And another question, if I may, please, on your graph of the specificity of the venom versus various species. Did you test versus human cell receptors?
Yes, we did. And they were also hit just as hard as the rest.
Okay, thank you.
Yeah, no worries.
I might just jump in just to, because I know time's getting towards the end now. So I guess I just wanted to, from behalf of the ACT Herpetological Association and the Canberra Reptile Zoo, thank both Christina and Chris for such a rich talk tonight, 360-degree talk. Lots of clapping, which just covered so many different issues from biology to ecology and even that kind of social dimension of attitudes towards these creatures. So we really appreciate you guys giving us your time. We know how busy you are, and we're really sorry, obviously, about the start, but we got there in the end. So that's a learning curve for everybody. But yeah, just thanks so much. And we're really excited to read some of these papers coming out and obviously just to keep the conversation going. So thanks to both of you guys for giving your time and your amazing insights.
No worries.
Thank you
Our
for
pleasure.
hosting us. And thanks for everyone's work improving our knowledge and positive sort of perspective on snakes in society. So we all have a part, and thank you everyone for doing that.
Thanks, guys. Cheers.
All right.
Awesome.
We've recorded that as well. Thank you.
Yeah, good on you.
Nice.
Okay.
Thanks, Christina. So we'll be in
I'll
touch
stop
about
sharing.
the recording, and yeah, sorry
All right.
for all the problems at the beginning. But,
Can't unsee it. All right. Thanks, guys.
No worries, guys.
Take care.
okay.
Good on you, Devin.
See you later.
Thanks, Jeff, Margaret. See you guys.